Have you ever stopped to think about your skin? I mean, really think about it. It seems so simple, right? Just a covering. But here's where it gets really interesting. It's actually your largest organ, a living, breathing, incredibly dynamic system. It's doing far more than you probably realize every single second.
It really is. And today we're taking a deep dive into that remarkable system, the integumentary system, our guide. The incredibly detailed insights from Histology, a Texan Atlas, the 8th edition. Our mission really is to cut through the complexity. We want to pull out the most important nuggets of knowledge. We'll be focusing on its microscopic structure, the key cells involved, the tissue types, and, what that all means for your health, the clinical side of things.
Yeah, think of this as your personal shortcut. You get to be truly well-informed about the skin. We're hoping for lots of surprising facts and those aha moments, stuff that gives you a whole new appreciation for your own body. Okay, so let's kick things off with the skin's grand design, the big picture, the integumentary system. It's truly massive. We're talking, what, 15% to 20% of your total body mass?
That's right. It's huge.
And it includes not just the skin itself, but also its derivatives like your hair, nails, and various glands.
Exactly. And this massive organ is organized into two primary layers. On the surface, the part you see and touch, that's the epidermis. This is your outer shield, a protective layer that's, interestingly, constantly renewing itself. What's fascinating is how it maintains a consistent thickness. It does this by shedding old cells, a process called desquamation, while new cells grow from below.
Okay, so constantly shedding, constantly rebuilding.
Precisely. And beneath that protective epidermis lies the dermis, this deeper layer. It's made of dense connective tissue. It basically provides the skin's strength, its elasticity, and its overall thickness, like the strong, flexible scaffolding holding everything up.
Right, the support structure. And even deeper, beneath the dermis, there's another layer, though maybe not technically skin.
Correct. That's the hypodermis. It's distinct but closely associated. Yeah. This is where your body stores fat, adipose tissue, organized into these little lobules. It acts as a major energy reserve and provides vital insulation.
Ah, so it's thicker in folks who are well-nourished or live in cold places, like a natural built-in coat.
Exactly. It's your body's natural padding and insulation layer.
Okay, so layers make sense, but what about functions? Why is skin so important, besides just covering us?
Well, because of its external position, it performs several essential functions. First and foremost, it's an incredibly robust barrier. Think about it. It protects you from physical impacts, harmful chemicals, microscopic invaders like bacteria and viruses. Plus, it guards against harmful UV radiation and prevents too much water from just evaporating out of your body.
So a mechanical, chemical, biological protection, and permeability barrier too. Wow.
But that's not all. Your skin is also a key player in your immune system. It actively captures potential threats, antigens, and signals your lymphatic system to respond. It's like an early warning system.
Immune surveillance right there on the surface.
Yep. And it helps maintain your body's internal balance, homeostasis, regulating temperature, preventing excessive water loss.
Right. Sweating to cool down, goosebumps when cold.
Yeah.
That kind of thing.
Precisely. It's also packed with nerves, providing rich sensory information, constantly relaying data about touch, temperature, pain to your nervous system. And surprisingly, it has endocrine functions. It helps convert precursor molecules, like those needed for vitamin D, into their active forms.
Vitamin D from sunlight, yeah.
And finally, it functions in excretion, helping get rid of waste products through sweat and other gland secretions.
That barrier thing. It's interesting how medicine uses that, like those patches, nicotine patches, motion sickness ones.
Exactly. That's a perfect example. It shows that while the skin is a fantastic barrier, it's also selectively permeable. We can use that property to deliver certain medications slowly and steadily right through the skin. It's clever pharmacology.
Okay, you hear people talk about thick skin versus thin skin. What's the actual difference there? Is it just thicker?
Good question. It's important to clarify that this distinction refers only to the thickness of the epidermis, that top layer. So your palms and soles, they endure a lot of friction, right? So they have a much thicker epidermis, and they're hairless. That's thick skin. Most of the rest of your body has thin skin, meaning a thinner epidermis, and it usually has hair follicles.
Ah, okay, so it's just the epidermis thickness.
Right. But here's a fun fact. The thickest overall skin, including the dermis, is actually on your upper back, even though its epidermis is relatively thin.
Huh. That is surprising. So when you really start to connect all these dots, the layers, the functions, the constant renewal, you really appreciate how crucial your skin is to simply existing. It's an amazing piece of biological engineering.
Absolutely. A true marvel.
Thanks for being part of the last-minute lecture family. Now, let's zoom in even closer, because here's where it gets really intricate, right? If we look at the epidermis, especially in thick skin areas like your palms, we can see 5 distinct layers, or strata.
Yes. Think of the epidermis as this dynamic factory. Cells are constantly changing as they move upwards. At the very bottom, resting right on the dermis, is the stratum basale, sometimes called the germinativum. It's basically a single layer of small, cuboidal or low columnar cells. These are the stem cells, the keratinocytes.
The ones doing all the dividing?
Exactly. They're mitotically active, constantly producing all the new cells for the layers above. The foundation, really.
Okay, so they divide, then what? They get pushed up.
They get pushed upwards into the next layer, the stratum spinosum, the spinous or prickle cell layer. Here, the keratinocytes are larger. They start developing numerous little cytoplasmic processes or spines. These connect to neighboring cells via structures called desmosomes.
Desmosomes, those are like cellular rivets, right, holding things together.
Perfect analogy. They link the cells tightly, giving the epidermis its strength and resistance to abrasion. This is where the cells start really building up their internal structure, synthesizing keratin filaments.
Got it. Strength layer. What comes next?
Next up is the stratum granulosum. It's usually just 1 to 3 cells thick,
Right.
but it's where a critical transformation happens. These cells start producing distinct granules. You can see them under the microscope. They contain proteins important for packaging keratin, but also, crucially, specialized lipids.
Lipids, like fats.
Exactly. These lipids get released and act like a waterproofing sealant between the cells. This forms a key part of your skin's water barrier.
Ah, the waterproofing layer. Makes sense.
Then, only in thick skin palms and soles you find the stratum lucidum. It often looks clear or lucid under the microscope. It's basically a transition zone. The cells here are becoming highly keratinized. They're losing their nuclei and organelles, packing tightly together, preparing for the final layer.
Like a final hardening step just for those high-wear areas?
Precisely. And finally, at the very surface is the stratum corneum. This is the outermost layer you interact with, composed of flattened, dead nucleate cells. They're essentially just bags filled with tough keratin protein. These are the cells that are constantly shedding.
The ones that flake off.
Yep. Its thickness varies a lot across your body. That's the main difference between thick and thin skin. And if an area gets increased friction, like on your feet from shoes, this layer thickens up. That's how you get calluses. Your skin's clever way of adding protection.
Right, calluses, extra armor. It's truly incredible, this constant differentiation and turnover. You said it's a specialized form of cell death, apoptosis.
It is, but a very specialized form. Instead of fragmenting and being cleaned up, these cells just fill with keratin, flatten out, and eventually detach. It's an incredibly efficient built-in renewal system.
How long does that whole process take, from a cell being born at the bottom to flaking off?
It's quite precise.
Yeah.
From basal cell division, it takes about 31 days for a cell to move up through the spinosum and differentiate into a granular cell. Then crossing that final stratum corneum layer takes another 14 days or so.
So total turnover, about 47 days, almost 7 weeks.
Roughly, yes. Around 47 days for the whole journey.
And that timing is important clinically, right? You mentioned psoriasis earlier.
Exactly. In psoriasis, that turnover time is drastically sped up. We're talking maybe 8 to 10 days instead of 47. This rapid production means cells pile up too quickly, leading to that increased epidermal thickness and the characteristic silvery scales you see. The shedding process just can't keep up.
And the shedding itself, desquamation, that's not just random flaking, is it? It's regulated.
Highly regulated.
Yeah.
It involves specific enzymes, proteases, that basically unzip those desmosomes, the rivets holding cells together. Near the skin surface, the pH becomes slightly acidic. This acidity activates these enzymes, allowing the oldest cells to detach cleanly.
Wow, even the shedding is carefully controlled by pH.
And when that control goes wrong, for instance, in Netherton syndrome, a genetic disorder, the body lacks a key inhibitor for those unzipping enzymes.
So they just keep unzipping.
Pretty much. It leads to uncontrolled desquamation. The skin barrier weakens, causing redness, scaling. It really highlights how vital that balance is.
Okay, and you mentioned the water barrier earlier, formed in the stratum granulosum. How exactly does that work? It sounds critical.
Absolutely essential for life on land, really. Keeps water in, keeps harmful stuff out. It's formed by 2 key elements working together. First, inside the cell, there's the cell envelope, CE. It's a tough layer of cross-linked proteins just under the cell membrane. Provides mechanical strength. Second, outside the cell, attached to the surface, is the lipid envelope. This is that layer of lipids like ceramides. It forms a sort of Teflon-like waterproof coating.
Teflon coating, I like that. Easy to picture.
And those lipids, they come from those tiny lamellar bodies we mentioned in the stratum granulosum. They basically discharge their lipid contents into the spaces between the cells, forming that continuous waterproof sheet.
And if that barrier gets compromised, I imagine that's bad news.
Very bad news. For instance, certain dietary deficiencies, like lacking essential fatty acids, can make the skin's barrier more permeable. Leaky, essentially.
Okay.
And even more dramatically, think about severe burns. They destroy this epidermal water barrier. That leads to massive, life-threatening fluid loss from the body. It really underscores how critical this seemingly thin layer is for survival.
A dramatic reminder of its importance.
Thanks for being part of the Last Minute Lecture family.
Okay, so we've covered the main skin cells, the keratinocytes, in detail, but you mentioned there are others in the epidermis.
Indeed. Beyond the keratinocytes, which make up the vast majority, the epidermis houses a fascinating array of other specialized cells, each with vital roles.
Let's unpack those. Who's up first?
Let's start with melanocytes. These make up maybe 5% of epidermal cells. They're derived from the neural crest during development and are found scattered among the basal cells right at the bottom layer. They look sort of dendritic, like little octopuses, but they don't form those strong desmosome connections with the keratinocytes.
And their job is making melanin, the pigment.
Precisely. They produce melanin pigment inside specialized organelles called melanosomes. And melanin is absolutely critical for protecting our skin cells, especially their DNA, from damaging UV radiation.
How does the melanin get from the melanocyte to the other skin cells? Does it just ooze out?
Not quite. It's a really cool process called pigment donation. The tips of the melanocyte's dendritic processes, which are packed with melanosomes, actually get phagocytosed, basically eaten by the surrounding keratinocytes.
So the skin cells eat the pigment packets?
Essentially, yes. The melanosomes then travel within the keratinocyte and arrange themselves over the nucleus, like tiny dark umbrellas. This physically shields the cell's precious DNA from UV light.
That explains tanning. More UV, more umbrellas needed. What about different skin colors? Do different races have different numbers of melanocytes?
That's a common misconception. Actually, all races have roughly the same number of melanocytes. The variation in skin color comes down to the activity of the melanocytes, how much melanin they produce, and especially how quickly that melanin is broken down by the keratinocytes.
So in lighter skin, the melanin breaks down faster.
Generally, yes. There are also different types of melanin, like eumelanin, brown-black, and pheomelanin, reddish-yellow, which contribute to the diversity of skin and hair colors. And other things influence pigment, too. UV radiation obviously triggers tanning. Certain hormones can cause darkening, like in Addison disease, and genetic defects can lead to conditions like albinism, where melanin production is faulty.
And clinically, melanocytes are important because of melanoma, right?
Exactly. Malignant melanoma. It's the most serious form of skin cancer because it originates from these pigment-producing cells and can spread aggressively. That's why checking your skin is so important. Remember the ABCD rule for suspicious moles. Asymmetry one half doesn't match the other. Border irregularity edges are ragged or blurred. Color variation, different shades of brown, black, sometimes red, white, or blue, and diameter greater than 6 millimeters, about the size of a pencil eraser.
ABCD. Got it. Early detection is key there. Okay, who's next in the epidermal cast of characters?
Next up are Langerhans cells. These make up about 2 to 5% of epidermal cells. These are actually immune cells, part of your body's defense system. They're bone marrow-derived, antigen-presenting cells.
So like guards patrolling the skin.
Exactly like that. There are dendritic cells, constantly surveying the epidermal environment for foreign invaders or antigens. When they encounter something suspicious, they capture it, process it, and then migrate out of the epidermis to nearby lymph nodes. There, they present the antigen to T lymphocytes, basically telling the immune system, hey, look what I found, we need to react to this, or sometimes, this is okay, stand down, inducing tolerance.
Immune initiation right in the skin. Interesting. Can you see them easily?
Not really in standard H&E stains.
Yeah.
They have a characteristically indented nucleus, and under an electron microscope, they possess these unique Birbeck granules that look like tiny tennis rackets. They're heavily involved in things like allergic skin reactions, contact dermatitis, poison ivy, that kind of thing. And unfortunately, they can also serve as a reservoir for viruses like HIV.
Okay, melanocytes for pigment, Langerhans for immunity. Who's the third type?
The third type is Merkel's cells. These account for maybe 6–10% of epidermal cells. They are also found in the stratum basal, that bottom layer. Their exact origin is still a bit debated, but they seem to have both epidermal and neural characteristics. They are most abundant in areas highly sensitive to touch, like your fingertips, lips, maybe the palms.
And their function is touch reception.
Yes, they are sensitive mechanoreceptors. They are closely associated with the flattened endings of afferent nerve fibers. The Merkel cell plus its nerve ending together form a Merkel corpuscle. They help you detect fine touch, texture, and sustained pressure.
And clinically, any concerns there?
Unfortunately, yes. A rare but highly aggressive skin cancer called Merkel cell carcinoma, MCC, can arise from these cells.
So below all this cellular activity in the epidermis, we have the dermis, the support structure. How does it connect?
Good question. The epidermal-dermal junction isn't just a flat boundary. It's quite uneven. The dermis projects upwards in these finger-like bumps called dermal papillae, which interlock with downward projections of the epidermis called epidermal ridges.
Like puzzle pieces fitting together.
Exactly. This creates a much larger surface area for connection, significantly strengthening the attachment between the two layers. It helps resist friction and shearing forces, especially in thick skin like hands and feet. Plus, specialized anchors, called hemidesmosomes, further bolt the epidermis down.
And the dermis itself has layers too, right?
It does. The more superficial layer, right underneath the epidermis and forming those papillae, is the papillary layer. It's made of loose connective tissue. It's rich in capillaries for nourishing the epidermis, lymphatic vessels, and lots of sensory nerve endings are concentrated here.
So lots of blood supply and nerves close to the surface.
Yep. Then deep to that is the reticular layer. This is considerably thicker and much denser. It's composed of dense, irregular connective tissue, meaning thick bundles of type I collagen fibers arranged in a sort of meshwork, along with coarser elastic fibers. This gives the skin its tensile strength and elasticity.
And those Langer lines you mentioned earlier for surgeons, are they in this layer?
Yes, the orientation of these collagen and elastic fiber bundles creates those lines of tension, the Langer lines. Incisions parallel to them tend to heal with less scarring because there's less tension pulling the wound edges apart.
This intricate structure, it really makes you wonder how all these cells and layers weave together. How do they provide not just protection, but also our incredibly detailed sense of touch? It's quite the system.
Thanks for being part of the Last Minute Lecture family.
Okay, let's unpack that sensory world a bit more. Our skin is more than just a barrier. It's this incredibly sensitive organ packed with specialized nerve endings connecting us to everything around us.
Absolutely. The skin's nerve supply is quite complex and varied. The most numerous type are the free nerve endings. These are basically bare nerve endings that extend right up into the epidermis, sometimes as far as the stratum granulosum. They don't have a fancy capsule or wrapping. They're like generalists, subsuming multiple senses: fine touch, heat, cold, and importantly, pain.
So the basic feelings.
Right. Some also wrap around the base of hair follicles, making you sensitive to even slight hair movements, like feeling a breeze ruffle the hair on your arm.
And we already mentioned the Merkel corpuscles with the Merkel cells.
Correct. Those Merkel cell neurite complexes are specialized mechanoreceptors, particularly good at sensing sustained pressure and texture.
What about the ones deeper down, the encapsulated ones?
Ah, yes. Deeper in the dermis and even the hypodermis, you find several types of encapsulated nerve endings. They have connective tissue or Schwann cell wrappings. First, the Pacinian corpuscles. These are quite large ovoid structures. Under the microscope, they famously look like a sliced onion.
A sliced onion, why?
Because they have many concentric layers, or lamellae, of flattened Schwann cells surrounding a central nerve axon. They're exquisitely sensitive to pressure changes and vibrations. You have lots in your fingertips, joints, even deeper tissues. They help you feel vibrations, like from a tool or a musical instrument.
Okay, Pacinian for vibration. What else?
Then there are Meissner's corpuscles. These are touch receptors, particularly responsive to light touch and low-frequency stimuli like flutter or stroking. You find them primarily within the dermal papillae, especially in hairless skin lips, palms, soles, fingertips. They help with fine tactile discrimination like reading braille or distinguishing textures.
So Meissner's for fine touch and texture.
And finally, we have Ruffini's corpuscles, sometimes called Ruffini endings. These are somewhat simpler encapsulated mechanoreceptors, elongated and spindle-shaped. They are sensitive to skin stretch and torque. They respond when the collagen fibers around them are displaced.
Stress receptors. So they tell you when your skin is being pulled or twisted?
Exactly. Like when you grip an object firmly or when your joints move, stretching the overlying skin. They provide information about sustained pressure and shear forces.
Understanding this intricate structure, especially the layers and cell types, becomes really vital when dealing with things like skin cancer.
Yep.
Precision is key, right? Which brings us to that surgical technique you mentioned, Mohs.
Yes, Mohs micrographic surgery, or MMS. It's a truly remarkable technique developed by Dr. Frederick Mohs specifically for removing certain skin cancers like basal cell carcinoma and squamous cell carcinoma and sometimes melanoma.
How does it work differently from standard excision?
The key difference is microscopic control during the surgery. The surgeon excises the visible tumor, but then, instead of just closing up, they take that tissue, map it meticulously with colored inks for orientation, and immediately process it in the on-site lab. They cut horizontal frozen sections, like slicing layers off the bottom of the excised tissue, stain them, and the surgeon, acting as the pathologist, examines these sections under a microscope right then and there.
So they check the edges immediately.
Exactly. They examine 100% of the peripheral and deep margins in one horizontal plane. If they see any remaining cancer cells at an edge, they know precisely where to go back and take a tiny bit more tissue only in that specific spot. They repeat this process layer by layer until all the margins are clear of cancer.
Wow, so it guarantees complete removal while saving the maximum amount of healthy tissue.
That's the major advantage: superior cure rates and maximum tissue conservation. It's especially valuable on the face, eyelids, nose, ears, areas where preserving tissue is critical for function and appearance.
Okay, that makes sense. Now, shifting gears slightly, what about the other structures embedded in the skin? The appendages, hair, glands, nails.
Right, the epidermal skin appendages. These are structures that develop as downgrowths of the epidermis during embryogenesis. Let's start with hair follicles in hair. You find hair almost everywhere except palms, soles, lips, and a few other specific spots. Each hair grows from a hair follicle, which is essentially an invagination of the epidermis that dips down into the dermis, sometimes even the hypodermis. The follicle has different regions, but the key part for growth is at the bottom, the hair bulb. This bulb surrounds a little nubbin of dermal tissue called the dermal papilla.
And the cells that make the hair are in that bulb.
Yes, the cells covering that dermal papilla form the hair matrix. These are rapidly dividing cells that differentiate to form the hair shaft itself and its inner root sheath. Melanocytes are also here, donating pigment to give the hair its color.
What's the hair shaft itself made of?
Hair is primarily composed of hard keratin, which is tougher and more cross-linked than the soft keratin of the epidermis. It typically has 3 layers: a central medulla, often absent in fine hairs. The main bulky cortex determines texture, elasticity, color, and an outer protective cuticle made of overlapping scale-like cells.
Like shingles on a roof?
Good analogy, yes.
Oh.
That cuticle protects the inner cortex.
And what's that little muscle that makes your hair stand on end?
Ah, the erector pili muscle. It's a tiny bundle of smooth muscle that attaches from the deeper part of the follicle to the superficial dermis. When it contracts due to cold, fear, or other stimuli, it pulls the hair follicle upright, causing the hair to stand on end and creating gooseflesh or goosebumps.
You mentioned stem cells earlier.
Oh.
Are there important ones in the hair follicle?
Absolutely critical ones. In a specific region of the outer root sheath, called the follicular bulge, there's a niche of epidermal stem cells. Under normal conditions, these stem cells provide the progenitor cells for hair growth and also contribute cells to the sebaceous gland. But what's really amazing is their role in wound healing. If you have a significant skin injury, like a burn that destroys the surface epidermis, these bulge stem cells can migrate out, proliferate, and help resurface the wound. They're a vital reserve for skin regeneration.
Wow, so hair follicles are like little reservoirs of healing potential.
They really are. And hair growth itself isn't continuous, it's cyclical. There's a growth phase called anagen, which can last for years on your scalp. Then a brief transitional phase, catagen, where growth stops. And finally, a resting phase, telogen, where the follicle shrinks and the hair eventually sheds, making way for a new anagen hair.
So not all hairs are growing at the same time.
No, thankfully. On your scalp, over 80% of hairs are typically in the anagen phase at any given time. This cycle is why things like laser hair removal require multiple sessions. The laser primarily targets pigmented hairs in the active anagen phase.
Ah, that makes sense. Targeting the active ones. From feeling that tiny breeze moving a hair to understanding how these follicles act as stem cell reservoirs, it really showcases the skin's incredible adaptability and function.
It certainly does.
Thanks for being part of the last-minute lecture, family. Okay, let's unpack this next part. Beyond hair, our skin is home to a variety of glands, each playing critical roles from moisturizing to cooling us down.
Absolutely. Let's dive into the glands of the skin. First up, the sebaceous glands. These are almost always associated with hair follicles. Together, the follicle, hair shaft, erector pili muscle, and sebaceous gland form what's called a pilosebaceous unit.
The whole hair and oil gland complex.
Exactly. You find the highest density of these on your face, scalp, chest, and back. though some, like meibomian glands in the eyelids, open directly onto the skin surface. These glands develop as outgrowths from the hair follicle's external root sheath.
And they produce?
They produce sebum. It's an oily, waxy mixture composed mainly of triglycerides, wax esters, squalane, and cholesterol. The way they secrete sebum is quite dramatic. It's called holocrine secretion.
Holocrine, what does that mean?
It means the entire secretory cell, the sebsite, fills up with lipid droplets. Then the cell itself undergoes programmed cell death, apoptosis, and disintegrates, releasing its entire contents, including the sebum and cellular debris, into the duct.
Wow, the whole cell sacrifices itself to release the oil.
That's the essence of it. New sebocytes are constantly produced by mycosis of basal cells at the periphery of the gland to replace the ones that are lost. Their activity is heavily influenced by hormones, especially androgens, which stimulate production, and estrogens, which tend to inhibit it.
Yeah, and sebum's function, just lubrication.
Lubrication and moisturizing the hair and skin is part of it. It also contributes to the skin's barrier function and gives each individual a unique chemical signature. Other potential roles like UV protection, immune modulation, or even pheromonal communication are still debated in humans.
And the clinical connection here is pretty obvious for many people, right?
Very much so. Acne vulgaris. This incredibly common condition is a chronic inflammatory disease of those pilospaceous units. It typically involves excessive sebum production, seborrhea, abnormal keratinization blocking the follicle opening, and the proliferation of bacteria, particularly Propionibacterium acnes, leading to inflammation, conones, blackheads, whiteheads, papules, pustules, and sometimes cysts.
Okay, so sebaceous glands for oil. What about sweat glands?
Right, sweat glands. There are two main types. The most numerous and widespread are the eccrine sweat glands. You have millions of these all over your body surface, except for a few places like the lips and parts of the genitalia. Highest density is on palms, soles, axillae, armpits, and forehead. There are simple coiled tubular glands that are independent structures. Their ducts open directly onto the skin surface via a sweat pore.
And their main job is cooling us down.
Exactly. Their primary role is thermoregulation. They secrete a watery hypotonic sweat. As this water evaporates from your skin surface, it takes heat with it, effectively cooling your body. They also slightly increase friction on palms and soles, aiding grip, and they act as an excretory organ, eliminating some waste products like salts, urea, and ammonia in the sweat.
How is the sweat actually pushed out?
The secretory part of the gland, deep in the dermis, has specialized contractile cells called myoepithelial cells wrapped around it. When stimulated, these cells contract and squeeze the sweat rapidly up the duct to the surface. Interestingly, these glands are controlled by the sympathetic nervous system, but paradoxically, they use acetylcholine as their neurotransmitter, which is usually associated with the parasympathetic system.
A little quirk of the nervous system.
A classic one.
Yeah.
And they respond to both heat, thermoregulatory sweating starts on the forehead scalp, and emotional stress. Emotional sweating often starts on palm soles axillae.
Any clinical correlations for eccrine glands?
A key one is for diagnosing cystic fibrosis. Patients with CF have a defect in chloride transport, leading to abnormally high levels of sodium and chloride in their sweat. The sweat test is a standard diagnostic tool. Also, in severe kidney failure, uremia, urea concentration in sweat can be so high that crystals form on the skin as the sweat evaporates, sometimes called urea frost.
Okay, eccrine are the main cooling ones. What's the other type?
The other type is epicrine sweat glands. These are much more limited in distribution, found mainly in the axillae, the areola of the nipples, the skin around the anus, and the external genitalia. Ceruminous glands producing earwax and the glands of Moll in the eyelids are considered modified epicrine glands. Structurally, there are larger tubular glands, often branched, and their ducts typically empty into the upper part of hair follicles, not directly onto the skin surface.
And their secretion is different.
Yes, they produce a more viscous, milky secretion that's rich in proteins, lipids, and carbohydrates. Initially, this secretion is odorless. However, when bacteria on the skin surface metabolize these components, it produces the characteristic body odor associated with these areas.
Ah, so it's the bacteria acting on the sweat, not the sweat itself that smells initially?
Precisely. And despite their name, epicrine, which suggests pinching off bits of the cell, evidence points towards secretion primarily occurring via the more standard merocrine process, exocytosis, similar to eccrine glands. Myoepithelial cells are also present to help expel the secretion. These glands become functional at puberty, influenced by sex hormones. Their activity is primarily linked to emotional stimuli stress, fear, pain, sexual arousal rather than heat regulation. They are thought to function as scent glands, possibly producing pheromones in humans.
And excessive sweating, is that related to these?
Pathological excessive sweating, hyperhidrosis, can involve either eccrine or apocrine glands or both. It's sweating beyond what's needed for normal thermal regulation, and it can be quite distressing.
Okay, glands covered. Now let's quickly touch on nails. We often take them for granted, but they're pretty specialized structures.
They are indeed. Nails are essentially plates of hardened, keratinized cells containing hard keratin. They sit on top of and are strongly attached to the underlying nail beds, which are specialized areas of epidermis. The proximal part of the nail root, is buried under a fold of skin. This root covers the nail matrix.
The matrix, that's where the growth happens.
Exactly. The nail matrix is the germinative zone. It contains stem cells that continuously divide, differentiate, and produce the hard keratin that forms the nail plate. Melanocytes, Merkel cells, and Langerhans cells are also present here. The hard keratin in nails is different from the soft keratin in your epidermis. It's more densely packed, has a higher sulfur content, and importantly, it doesn't desquamate, it just grows continuously outwards.
That crescent moon shape at the base, the lunula? What's that?
The lunula is the visible part of the nail matrix, usually whitish because the underlying matrix is thick and obscures the pink nail bed underneath. It represents the distal zone of active keratinization. The epimysium, or cuticle, is the fold of skin overlapping the nail root. And the hyponychium is the thickened skin under the free edge of the nail plate, sealing it to the fingertip.
How fast do they actually grow?
Fingernails grow roughly 2–3 millimeters per month, while toenails are slower, maybe 1 millimeter per month. That means complete replacement takes about 6 months for a fingernail and up to 18 months for a toenail.
This raises an important question. With all these intricate parts, layers, cells, appendages like follicles and glands, how does the skin manage to heal itself when injured? It seems like a complicated repair job.
It is complicated but remarkably efficient. Skin repair, or wound healing, is a fascinating process. We generally talk about 2 types of healing. Primary union, or first intention, occurs in clean surgical incisions where the edges are precisely brought together. Healing is relatively quick with minimal scarring. Secondary union, or secondary intention, happens in larger, more traumatic wounds where the edges are separated and there's significant tissue loss. This involves more inflammation, the formation of extensive granulation tissue, new connective tissue and capillaries, and often results in more noticeable scarring.
What actually happens in the dermis during repair?
First, a blood clot forms to stop bleeding and provide a temporary scaffold. Then immune cells like macrophages move in to clean up debris and damaged tissue. Fibroblasts proliferate and migrate into the area, producing new collagen and extracellular matrix to form granulation tissue. Some fibroblasts differentiate into myofibrils, which contain contractile proteins and help pull the wound edges together. That's wound contraction. Eventually, this granulation tissue matures and remodels into scar tissue.
And the epidermis, how does it close the gap?
Basal keratinocytes from the healthy skin at the wound edges start to proliferate and migrate across the surface of the granulation tissue underneath the scab. They basically crawl across to meet in the middle and restore the epithelial barrier.
And those stem cells in the hair follicles?
They are crucial, especially for larger wounds. If the entire epidermis is lost over an area, like in a deep abrasion or a second-degree burn, stem cells residing in the hair follicles and sweat glands within the wound bed can migrate out and contribute significantly to re-epithelialization. If even these structures are destroyed, like in a full-thickness third-degree burn, then spontaneous re-epithelialization can't happen, and skin grafting becomes necessary.
So the appendages really are vital reserves for healing.
Amazing. And that brings us to the end of our deep dive into the fascinating glands and nails of the integumentary system and the remarkable process of skin repair.
Thanks for being part of the last-minute lecture family. Wow, what an incredible journey through the integumentary system. We've really unpacked how the skin's layers, those specialized cells, and all the unique appendages work together, from protecting us against UV rays and microbes to letting us feel the world around us and even healing itself. It's truly a testament to biological ingenuity.
It really is. And if we connect this to the bigger picture, considering the skin's incredible dynamic equilibrium, its vast network of sensory input, and especially those stem cell niches hidden away in structures like hair follicles, it really raises an important question for the future, doesn't it?
Like what?
Well, what unexpected ways might medical science leverage the skin's inherent regenerative capabilities? Can we harness these processes for, say, widespread organ regeneration, or perhaps develop highly personalized drug delivery systems based on skin biology? far beyond what we imagine today.
Using the skin's own tricks for broader medical applications. That is provocative. Imagine the possibilities. Well, we hope this deep dive gave all of you plenty of those aha moments and definitely a new appreciation for the incredible organ that is your skin. Keep that curiosity alive.
It's been great sharing this deep dive with you all.
Join us next time for another deep dive into fascinating subjects.